friends, this is Sakshi. Today we are going to learn about the topic lag operon of class 12th chapter molecular basis of inheritance. So let's start. Lag operon. So guys, first of all, we should know that who were the scientists who discovered lag operon. So Franco is Jacob and Jacques Monod are the two scientists who discovered lag operon. So guys, we are going to learn about lag operon in prokaryotes. Prokaryotes such as bacteria. Let's know the difference between prokaryotes and eukaryotes. Eukaryotes are those who have well-defined nucleus. Here, the genetic material is stored, such as human, protista, each. Prokaryotes are those in which nucleus is not well-defined. The genetic material is spread all over it. Guys, here, one promoter regulates 3 genes, and this type of DNA sequences are called polycistronic, which is found in prokaryotes. Like that, there is one more term, monocistronic. Here, one promoter regulates 1 gene, and this type of DNA sequences are called monocistronic. Just opposite. Here, one promoter regulates 3 genes. Here, one promoter regulates only 1 gene. Guys, in lag operon, lag refers to lactose and operon refers to system. Means a system consisting of lactose. Guys, lactose matters a lot here. If lactose is present in the system, then the system is called on system. And if lactose is absent, then it is called off system. So we will see the first case where inducer is absent, means lactose is absent. Here are the genes. These are called Z, Y and A are called structural genes. O is called operator, P is called promoter here, and I is called regulatory gene. So what will happen here? Here transcription occurs and repressor mRNA forms. Further, there will be process of translation and repressor formation occurs. This repressor will bind with operator. Here, repressor protein is binds to the operator design and it prevents RNA polymerase from transcribing the operon. So here, no protein formation occurs, no transcription, no translation. System is called off system, inactive system. Now see the second case, where inducer is present, means lactose is present. Here what will happen? Regulatory gene I will transcribe repressor mRNA, means process of transcription occurs here and repressor mRNA forms. Now it will make a protein that is repressor, but here is a condition that inducer, that is lactose, is also indulged in this, by which the shape of this protein changes, something like that. For that reason, it is inactive. It is called inactive repressor. It will not bind with operator design here because its shape varies. Now this promoter gene promotes this all 3 for transcription, through which mRNA forms. Further, there is a process called translation occurs here. Z gene forms, I'll write here, Z gene forms beta galactosidase protein here. Y gene forms homease protein and A gene form trans acetylase. Guys, here beta galactosidase break lactose into glucose plus galactose. Permease permit or allows lactose inside the bacteria's body. Means permease permits lactose inclusion or inclusion inside the bacteria. And trans-acetylase function is still not known, but it is considered that it functions same as beta galactosidase. Guys, here we can use lactose or allolactose. Both we can use. I'm showing you this diagram of NCERT book. See here. In absence of inducer, there is no lactose, allolactose present. So what will happen? Regulatory gene transcripts and form repressor mRNA. Further translation occurs and repression protein will form. It will bind with operator. In this case, repressor binds to the operator design and prevents RNA polymerase from transcribing the operon. So there will be no transcription occurs. In the presence of inducer, what will happen? Regulatory gene will form repressor mRNA. Further, in inclusion of inducer or lactose, what will happen? The repressor will inactive, means its shape changes like that. So there will be no meeting of repressor with the operator. So promoter will promote all three structural genes for transcription. mRNA will form. Further, there will be a process called translation occurs, and Z gene codes for beta galactosidase, Y gene codes for permease, A gene codes for transacetylase. So beta galactosidase will break lactose into glucose and galactose. Permease will permeate lactose into the bacteria body. Transacetylase, removal of beta galactose toxicity. So this is the operon, lac operon.